GeneCard suite development in the Lancet lab. And those of you who heard the PubMed talk this morning, I was wondering if I would type Lancet into the new PubMed system, if I'd come up with the journal or their own Lancet, and I'll have to definitely try that later. And so I'm going to talk about how when you have whole genome sequencing, you have an avalanche of data, most of it in the non-coding regions. So we, in the GeneCard suite, we have a very rich environment and knowledge base of human gene disease associations when we enhance them with regulatory elements, this genomic dark matter and non-coding RNAs. We want to be able to use and leverage all of this information together in order to decipher genetic diseases. So the talk outline is I'm going to give you a few words about the GeneCard suite at a glance, and then some introduction, small introduction about enhancers, regulatory elements, and non-coding RNAs very, very fast, and then take that information and talk about it in the context of our system, and we'll proceed from there towards the goal, of course, of deciphering the genetic diseases, doing a better job of that with this genomic dark matter. Okay, so how many here have used or heard about GeneCards in the audience? Just curious? Okay, great, that's wonderful. How about some of these other suite sites? Any of them, MalikCards, Varilect? Okay, that's what I'm afraid of. This, oh, great, one in the back. Okay, yeah, so I'm going to give you a few words about each of them. Whereas you know that GeneCards is gene-centric with a lot of annotations about a gene, your first step, one-stop shop to learn about a gene. MalikCards tries to do the same thing for disease information, your malady information, one web card for each disease, and it's responsible for the gene-disease relationship matrix, and we'll go into that in a little more detail. PathCards is pathway-centric. Life Map Discovery talks about embryonic development and stem cells. That won't be covered here. It's just another source of annotation, though, in the knowledge base. GeneLoc is chromosome-based, so if you want to look at all of the genes on a particular chromosome, you have that information. It's also responsible for unifying the main GeneCards gene symbol names from sources like Ensemble, NCBI, which sometimes have different names and identifier, and based on the location, it chooses what our GeneCards gene main symbol is and its aliases. On top of all that, we have a set of analysis tools, and maybe the next time the talk that will be given on Network Assessor talks about gene set enrichment analysis, maybe gene analytics will be included in that list along with David and Enricher, because given a set of genes, it will produce as output the annotations that are enriched along different axes, pathways. If they share pathways, share diseases. Our batch query engine, Gene a la carte, given a set of genes, you can choose your annotations and get the list of them. Related genes, genes like me, given a gene, find other genes that are similar to it. Say you can call it actual paralogs, functional paralogs that share a domain. That's genes like me. And the newest one, which is relevant to this talk, is GeneHanser, which is responsible for defining regulatory elements and their target genes. Now, all of this knowledge base is leveraged by our NGS analysis tools. Varilect is a NGS phenotyper. Given a set of genes and a phenotype of interest, it prioritizes the subset of the genes that are related to the phenotype, and I'll show you that in more detail. And TGEX is an end-to-end solution, starting with a VCF file and ending with the report, and I really won't talk about that all that much. Okay, so back to enhancers. So non-coding regulators, just at a glance, and actually this was mentioned in one of the previous talks of the conference. I'm sorry, I forgot who actually showed the picture of this many fingers polydactyly. It turns out that enhancers are distant-acting, non-coding regulatory elements, and there are many of them. They're like 400,000, which is tenfold if you compare it to the number of genes. And very often, they're involved in development and disease, and a mutation in a regulatory element, in an enhancer, can impact the gene itself. And in this example, you see in mice and in hands and in humans where you have truncated fingers. And that's all because of a mutation in an enhancer that's, let's see, how many kb? About a megabase away from the gene from the sonic SHH, sonic hedgehog gene of interest. So what GeneHanser does is it collects information and produces a database of enhancers. Okay. moving to non-coding RNAs, that other section, that other element of the dark matter that we're talking about, they've been increasingly acknowledged for their importance in development and in disease. And our project 5 years ago wanted to include non-coding RNAs in GeneCards. That's a while. There's a paper about it, but the time has come to upgrade. And now there are many new sources, RNA Central amongst others, which has RNA gene, ADBI, which has RNA genes transcript information. What we're doing is we're elevating this information, as we've done in the past, to actual gene-level RNA genes, including their information, their annotations, all towards the goal, again, of the topic of this talk, of being able to be leveraged in solving clinical cases, because sometimes they are very important in diseases and in development. Okay, so that's by way of introduction. And just a couple of words about GeneCards. I won't dwell on it because most of you are familiar. It has many visits. People like it. Comprehensive search engine, many different sections. Enough about GeneCards. Now, what MalikCards does is when we started to do this disease-based database, we said, fine, no problem. We'll find a list of disease somewhere out there. Maybe OMIM has it, maybe a different source has it. And then we'll use our strengths in finding other sources to annotate these MalikCards diseases and come up with disease-centric pages. It turned out, though, we couldn't find one that we were very happy about, but we did find about 15 that all were great. And we have heuristics in order to take we prioritize this list of disease name sources, and we canonicalize the names from the different sources, and we unify them where possible. We eliminate trivial words, of course, in the naming. We know that liver carcinoma and neoplasm of liver are probably the same disease. It gets more complicated than this as well, but let's say in a nutshell, we end up having right now about 19,000 disease names. And we collect the aliases, and as we map according to identifiers like disease ontology, identifiers ICD 9 and 10, we include all that in our main MalikCards disease page. And again, the hope is that more people will access. Actually, it has become more popular, even though maybe not in this room, for looking at it as a place to learn about diseases. It's responsible for the disease gene matrix, and we've come up with a concept of elite gene disease associations, which means that if the gene disease association are supported by manually curated and reliable sources, then we give an asterisk and we say this is an elite association, and when we're doing interpretation, this gets ranked higher when you're trying to rank and say how relevant is a gene to a phenotype. And some of these examples of that are causative mutations annotated by OrphanNet, OMIM's molecular basis known, the existence of a genetic test. Okay, but in addition to that, there are also non-elite associations that sometimes other sources don't have, where you have text mining that will give information relating the gene to diseases. And also, we leverage GeneCards. We search for the disease within all of the GeneCards information, and that also gives us inferred relationships in MalikCards. And then there are other sections where you have related diseases, Go terms, that we don't have time to go into. Okay, so what's the new stuff? Let's talk about GeneHanser. GeneHanser is a project that started just a little over a year ago, about a year and a half ago. And it's an integrated regulatory elements database where there are many databases, each with their own strengths and weaknesses. And so a PhD student, Simon Fischer-Levitz, in our groups, took on this project, starting with 4 sources of information about enhancers: ensemble regulatory build, ENCODE, Phantom, and Vista, and define clustering algorithms in order to come up with unified enhancers. And recently, we've added promoters into the mix, and sometimes the regulatory elements could be an enhancer or a promoter or both, and have that annotation. And again, the importance is that you have enhancers that are defined as elements in of their own right, and they could be very strong enhancers because they're remarked upon by many of the sources. But in addition to that, what's important to us is the enhancer-gene relationship, similar to that example that we saw, where if you have a mutation in an enhancer that impacts a gene, then you'll know to link that gene to the disease. And in a minute, later on at the end of the talk, we'll see an example of that, of where this was used to help solve a clinical case. In our pipeline, are trying to find more sources of information about enhancers, especially enhancer disease, and information, and we're working on that right now. And a few words about non-coding RNAs. It turns out that in GeneCards we do have about 67% of the genes are RNA genes, but it's based on not recent information, FRNA-DB, and we've decided to have the project to upgrade this information. And essentially what we're doing is that we're consolidating genes by using many new sources, very new work in progress. And the sources that are being looked at are RNA Central, RFEM, Phantom5, Mir2 disease, LinkRNA-DB, Stasher, CHESS, and also literature-based sources that we already have some of this information in MalikCards, literature-based, manually curated enhancer disease associations. And the idea is to upgrade the basis in the knowledge base to include as high-quality RNA gene information in the same way as we're including high-quality enhancer gene information towards the goal of solving more cases. There are some diseases like the RDICC disease that only has an RNA gene associated with it. That's already in the GeneCards knowledge base. Other diseases like diabetes has many elite, even non-elite protein coding genes associated with the various forms of diabetes. But in addition, there are many RNA genes that, while we're prototyping this project, we've come across that are not yet in our system, and they will be added as a result of this work. Okay, so I keep talking about deciphering genetic diseases, solving clinical cases. How do we do that? Okay, so the name of our tool is VariLect, for variant elect. The interesting thing is that the input to VariLect is actually a set of genes. The assumption is that the variants have been mapped to genes. The TGEX system that I'm not going to talk about actually takes care of that part of the job. What VariLect does is its job is to take the set of genes and look at the phenotype and see how can you find a subset of the genes that are matched best to the phenotype by leveraging all of the rich information in the GeneCards suite knowledge base, the gene, disease, pathways, and all those entities and the relationships amongst them, and return a ranked score. And I'll show it in action in a minute. We know of hundreds of cases that have been solved using VariLect, enabling accurate clinical diagnosis and just as important, proper genetic counseling so that a family will know if it makes sense to do PGD for the next pregnancy based on this information. And this is just a quick look of VariLect in action. This is the screen where you have an input set of genes. This is our sample. If you go into variLect.GeneCards.org, you will be able to see this as an example. If you just click on it, get a list of genes. This is actually a true case of a certain kind of diarrhea in infancy where the babies were not getting well. And by using VariLect and getting a scored and ranked set of genes, based on the information in the knowledge base, we were able to find a top-ranking gene. Oh, wait, sorry. Back. Okay, the gene was TTC3R7. The patient had this atypical syndromic congenital diarrhea. This top-scoring gene is a known gene for trichohepatoenteric syndrome, THES, and it led to the diagnosis. This was actually published, and this was found based on exome sequencing. So this is where VariLect was strong before we even got into the enhancer project. And now what we want to do is we want to make something strong even better. One thing that I wanted to mention about VariLect is, in addition to finding these direct relationships, there is a non-obvious way that VariLect also links your subset of your genes to their phenotype of interest. Sometimes gene A is directly related to phenotypes of interest. And note that the phenotypes that you can specify in VariLect are not just controlled vocabularies like HPO terms, but it could be any keyword at all that's could be in a paper, could be something in a name of a pathway, a Go term, anything, or a symptom, some information in a summary about a disease or about a gene if you wish. So often the direct mode matches it. We have a scored set of results. In addition to that, though, there's the indirect mode, and this is sort of similar in what was talking about in network assessor, but not exactly the same. What we come up with is a very sophisticated all against all gene-to-gene matrix, where you relate genes to each other based on all of the information in GeneCards. So if they share a protein-protein interaction, for example, if they're involved together in a protein-protein interaction, or if they are in the collective gene set of a particular pathway or superpath, which I don't really have time to talk about here, then by guilt by association, we'll say, okay, gene A is not connected to phenotype of interest, but gene A is connected to gene B, and therefore we can infer an indirect relationship between gene A and the phenotype. And often this finds results where the direct result doesn't find it. So, okay. So how does GeneHanser fit into the picture? So using exome sequencing in the example before and other examples, the coding variants are mapped to genes, and then the genes are running using the Verilect algorithm to connect them to the phenotype, and hopefully we'll get good results. With GeneHanser, with whole genome sequencing, with the myriad of variants, they take advantage of the GeneHanser system, which which produces regulatory elements, and the regulatory elements have the gene associations as well. This is a many-to-many relationship. All of this information is thrown into the mix, and then we could, thank you, and then we could solve the, then Verilect can hopefully solve the issue. And an example in the case study is this is a rare congenital genetic skin disease. This is in collaboration with Ofer Sarig and what's the other person? And Eli Sprecher, who's the physician at Soreski Medical Center in Tel Aviv. And this is a real case where they were trying to solve this rare congenital disease. And so it's a study of a large family with, it's called FFDD1, focal facial dermal dysplasia type 1. These FFDD star are just a group of developmental diseases that are related to characteristics of characterized by lesions on the face. And the first thing, of course, they tried was to look at the known disease-causing genes, because there are many different types of diseases in this family, and those were ruled out immediately. And it turns out that all of the patients had a duplicated 180 kb CNV shared. All the patients had it, and of course they thought that maybe there'd be a mutation in that CNV gene, but it turned out not to be the case. Verilect was used to analyze the genes, and because it had the enhancer information, it found that the mutation, that the variant, disease-causing variant, was actually within a target, was actually within an enhancer in the CNV region, okay, which was a target, and the gene of interest that Verilect found, MTOR, was a target of this enhancer with that mutation. And then GeneHanser gives its own identifiers, and it was 450 kb away from MTOR, but Verilect found this association, and this was confirmed by immunohistochemical staining, and they saw upregulation of MTOR. And we were very excited, and then they were able to present it at a medical conference. And so getting back to the original slide, I hope that I've convinced you by exploring the dark matter of non-coding RNAs and regulatory elements, enhancers and promoters, we're able to leverage the knowledge base in GeneCards even so that we can do an even better job of deciphering genetic diseases and finding the light bulb of which is the variant that is responsible for the disease of interest. And I'd just like to thank our team of people in our team at the Weizmann Institute, an interdisciplinary team of data scientists, software engineer, doctors, and quality assurance, together with Like Map Sciences that fund our research, who've developed TGIX as well based on our terms, on our knowledge base and suite. And I thank you very much.
Thank you. Thanks so much. Are there any questions, comments, thoughts, observations?
Yes.
Very nice talk.
Thank you.
For GeneCards for commercial user, kind of things to require license?
For commercial users, GeneCards requires a license. We're a small group, and even though it looks like we have many projects in the team, in GeneCards, we need to be able to fund our research, and they pay for our research. So yeah, LifeMap Science is the...
License requirement for other cards, like
It's free for academic use. If a commercial user wants to use GeneCards, then they have to get a license. Like Big Pharma use this information. They want it behind the firewall, for example, so that people don't know what genes they're studying. But you can try out anything. If you want to try out Varilek, there's no licensing. Even TJEX, yeah, you could try it, have a trial, and see how you like it. And we'd rather have people look at it and not pay than not look at it. But eventually, if you're a big pharma company and it's seen that you access the site too much, our partners will find them.
Okay, thank you.
You're welcome.